Hello everyone. This video we're going to discuss about phagocytes did phagocytosis. Okay, there's a very important concept that we have to know that phagocyte is the cells and phagocytosis is a process. This right here in biology terms is referred to a process. So what process? Process of destroys microbes. And this phagocytosis process occur in second line resistance in our body, in our human body. And know that in the second line of resistance, we're going to talk a lot about white blood cells. So this phagocyte is mainly, not mainly, it's this phagocyte is white blood cells, but there are specific white blood cells that did phagocytosis, which are neutrophil, and macrophage. All right. Okay, now that we got everything we need to know, let me ask you this question. How a phagocyte destroy microbes, or how this process of phagocytosis happened? Okay, let's check this out. Okay, how a phagocyte destroys microbes. The first step, or the first process that is happen, is detection. Here are a picture of neutrophil, or the phagocyte we talk about. Neutrophil. Here are the nucleus, and here are some microbes. So the phagocyte, or the neutrophil right here, detects microbes by chemicals they give off. So if there's a microbes inside our body, this neutrophil will detect it and will come towards the microbes. Okay, so phagocyte detects microbes by chemicals they give off, and phagocytes move toward detected microbes. Okay, the second one is ingestion. The microbes that has stuck to the surface of phagocyte is engulfed in by pseudopodia, and then it's going to form a vesicle inside the cell, inside the neutrophil. So we call this pseudopodia. Pseudopodia is a part of the phagocyte that is able to wrap microbes whenever they near one. Whenever the neutrophil is near a microbes. So it's going to engulf the microbes into the cell. Okay, engulfing microbes in and forming a vesicle. If you remember, what is a vesicle? Vesicle is an organelle inside the endomembrane system on a cell, inside a cell, that acts like a delivery man. Okay, that acts like a delivery man, which is transferring stuffs inside our cells. Okay, then the third one is phagosome forms. Of the forming of a phagosome. Phagosome is just a phagocytic vesicle. Okay, but what does phagocytic vesicle means? It's a vesicle that have a part in cycle, in phagocytic cycle, phagocytic process, you might say. So it's a vesicle that helped, that is used, that is a part of phagos phagocytic phagocytosis. Okay, here are the phagosome. Inside this a microbes that is stuck and cannot go outside. Okay, the next process is a fusion with lysosome. Phagosome fuses with lysosome, which is an organelle, again, an organelle inside our cells that contains a powerful enzymes that can digest those microbes. Okay, here are the zoom in part of this. The microbes and the enzymes going to fuse, and it's going to digest in the next process, digestion. Okay, here it says on digestion, the microbes are broken down by enzymes into their chemical constituents, or simply, microbes is teared apart by enzymes to an unfunctionable part. So let's have a look. Let's have a zoom in here. Okay, this was the structure of the microbes before it's broken down by the enzymes, and here are some the structures that already broken down by the enzymes. Okay, you can see it dissolving, not dissolving, disassembling apart from the original shapes. Okay, okay, the last process of phagocytosis is discharge. uh, Indigestible material is discharged, but which one is the indigestible material? On the last process, microbes is teared down by enzymes to an unfunctionable part. So the one that indigestible material is the unfunctionable part that doesn't need to broken down even more. Unfunctionable part that doesn't need to broken down even more. Okay, that's about it on phagocytosis, but I have a question for you to challenge yourself. Why did phagocytosis occur? Why did phagocytosis said to be occur in the second line of resistance?